Hello guys, welcome back. Today we're doing meiosis, and in order for you to be able to answer the questions under the topic of meiosis, you need to know what is meiosis. You need to know the process of meiosis in animal cells. You need to know the first meiotic division, which is meiosis 1. You need to know second meiotic division, which is meiosis 2. You need to know the significance of meiosis. You need to know abnormal meiosis. You need to know the differences between meiosis 1 and meiosis 2. At the end of this lesson, we will answer some exam questions. So, first thing, what is meiosis? Meiosis is a cell division of a diploid cell into 4 haploid cells, which develop to produce gametes. A diploid cell has two set of chromosomes. A haploid cell contains a single set of chromosomes. In human, gametes, which are haploid cells, contains 23 chromosomes. And a diploid cell contains 46 chromosomes. A gamete is an egg cell in females, and it's a sperm in males. This process of meiosis is a process where gametes are produced. Now, if they ask you a cell division where gametes are produced, your answer should be meiosis. But if they say a process where gametes are produced, that is gametogenesis. In females, it is oogenesis. In males, it is spermatogenesis. So the question can be specific. If they ask you a process whereby gametes are formed in females, the answer is oogenesis, and in males, it's spermatogenesis. In females, this process takes place in ovaries, and in males, in testes. Now, for us to have gametes, the process of meiosis has phases that I call PMET1 and PMET2. P for prophase 1, M for metaphase 1, A for anaphase 1, T for telophase 1, and P for prephase 2, M metaphase 2, A anaphase 2, and T telophase 2. So we have a process, the first stage of meiosis, where DNA replication takes place, and that is called interphase. This is where DNA replicates to form copies of itself. Before we go through the phases, let's look at a chromosome. This is what we call a chromosome, and when we have two of them like this, they are homologous chromosomes. But that's if they're equal in height and they have the same shape and the same size. We have a centromere here in the middle that holds the chromatids together. These are the chromatids. Now, during prophase 1, homologous chromosomes pair, and then a process called crossing over occurs. Now, this is how the process of crossing over occurs. The two chromatids of homologous chromosomes, they cross at a point called chiasma, and the two chromosomes exchange genetic material. They become genetically different. And the importance of crossing over is that it brings about variation. In this phase, prophase 1, spindle fiber also forms. In metaphase 1, the homologous chromosomes align themselves in the middle. In anaphase 1, homologous chromosomes separate, and in telophase 1, the chromosomes are enclosed in clay. We have two daughter cells. During this phase, we have division of cytoplasm, which is called cytokinesis. In prophase 2, a new set of spindle fiber forms. The chromosomes condense. In metaphase 2, the chromosomes, not homologous chromosomes this time, they align themselves in the middle of the equator. And in anaphase 2, the chromatids separate, not chromosomes, right? In telophase 2, 4 haploid daughter cells form, in which each chromosome has just one chromatid. Cytokinesis also takes place during this phase. Those 4 daughter cells are sperms in males, and in females, it is an ovum because only one of them becomes functional. Now let's talk about the significance of meiosis. There are 2 reasons why meiosis is important. It reduces the number of chromosomes by half. In other words, from diploid to haploid. This ensures that the sex cells have half the number of chromosomes of the other somatic cells so that when fertilization occurs, the zygote formed has the correct number of chromosomes. It balances the doubling effect of fertilization. Number 2, crossing over introduces genetic variation. Genetic variation results in offspring that are better adapted to a particular environment and ensures that they will have better chance of survival. Now let's look at abnormal meiosis. Sometimes mistakes occur during the process of meiosis. This can happen in anaphase 1, where the homologous chromosomes may not separate, also called non-disjunction. It can also happen in anaphase 2 when there is non-disjunction of the sister chromatids. If there is non-disjunction of chromosome pair 21 in humans, it leads to the formation of abnormal gamete with an extra copy of chromosome 21. If there is fusion between a normal gamete and abnormal gamete with an extra copy of chromosome 21, it leads to Down syndrome. Now let's look at the differences between meiosis 1 and meiosis 2. In meiosis 1, the chromosomes arrange themselves at the equator of the cell in homologous pairs. In meiosis 2, chromosomes line up at the equator of the cell individually. Whole chromosome move to the opposite poles of the cell. In meiosis 2, chromatids move to the opposite poles of the cell. In meiosis 1, 2 cells form at the end of this division. In meiosis 2, 4 cells are formed at the end of this division. In meiosis 1, the chromosome number is halved. In meiosis 2, the chromosome number remains the same. Crossing over takes place in meiosis 1, and crossing over does not take place in meiosis 2. Now let's look at the exam questions under this topic. The first question: the diagram below represents all the chromosomes in a cell that is undergoing normal cell division. Name the type of cell division that is occurring in the cell in the diagram. We know that we spoke about cell division, which is meiosis, right? So the answer is meiosis. B, name the phase of the cell division during which the chromosomes behave as shown in the diagram. If you look at the diagram, you can see that crossing over is taking place. So in which phase does crossing over take place? The answer is prophase 1. Where in human female body would the type of cell division named in question 1.4.1 take place? The process takes place in ovaries. Give the letter and name of the structure that attaches to the spindle fiber. During anaphase 1, centromere, which is letter C, it attaches to the spindle fiber, right? Number 1.4.3, give the letter and name. Point 4, how many chromosomes will be found in each daughter cell at the end of this cell division? The answer is 3. The diagram below represents a chromosome pair undergoing a process during meiosis. It's still crossing over here. Name the organ in the human male where meiosis occurs. The answer is testis. Name the process represented in the diagram. The process is crossing over. Number 5, 1.5.2, label area P. Area P is chiasma, structure Q, centromere, structure R, chromatid. The diagram below represents different phases of meiosis in an organism. Identify A, B, C, where is A? A is centromere, B is homologous chromosomes, C is spindle fiber. Identify the phase represented in diagram 3. Diagram 3 is anaphase 2 because chromatids separate. Write down the numbers of the diagrams to show the sequence in which the phases occur. Diagram 1, that is prophase 2. Diagram 2 is metaphase 1. Diagram 3 is anaphase 2. So the answer is 2, diagram 2, diagram 1, diagram 3. So 2.1.4, state one difference between metaphase 1 and metaphase 2. During metaphase 1, homologous chromosomes arrange themselves at the equator in homologous pairs. In metaphase 2, chromosomes line up at the equator of the cell individually. Thank you so much for watching. If you don't understand, please comment down below the comment section and I'll answer as soon as I can. Don't forget to like and subscribe. I would really appreciate it. Thank you.